Okay, good morning. It's my pleasure today to tell you a little bit about Bayesian variable selection. I'm going to try to keep this primer pretty informal, so please stop with questions if you have any questions. So the context of this primer is that of generalized linear regression, right? So we use linear regression and variants of it like logistic regression all the time in data analysis. So let's just set up some notation that we're going to use throughout this primer. So first of all, we imagine we have some data set of n data points. There's some covariate vector x, which is p-dimensional, and there's some response or outcome variable y, which, in the case of vanilla linear regression would be continuous, but it also might be, binary or some other count, right? So this is a pretty generic setup that we encounter all the time in data analysis. And one way this is sometimes depicted is at the bottom, right? We have some outcome y. We apply some link function f to a linear combination of our covariates, and there's some noise that we add at the end, right? So this sort of covers different cases, including linear regression, in which case the noise might be Gaussian, but it also sort of, in cartoon form, covers other cases like logistic regression. So everything I'm going to be saying throughout this primer is pretty generic, and it kind of applies to different variants of generalized linear regression. But for the majority of the following slides, I'm really going to just focus on the linear regression case because it's the easiest and simplest one to talk about. But all these ideas kind of generalize to generalized linear regression more broadly. Okay, so let's say we're doing generalized linear regression. What are some things that we might want our inference method to do in this context, right? So number one might be interpretability. In some ways, this is automatic in the sense that once you limit yourself to a relatively small class of models like linear regression, you automatically get at least some interpretability, right? Because each covariate x now has a coefficient beta that's associated directly with that covariate. And so if you infer that beta is, say, positive or negative, that's relatively easy to interpret in the context of your data. But we might, of course, want more than that, right? Like maybe not only do we want point estimates, but we also maybe want uncertainty estimates, or maybe we want rankings of the importance of different covariates. Is covariate 6 more important than covariate 12, and so on and so forth. So interpretability is sort of, there are many sort of facets to it that we might desire to have. Another thing that we might want to have is good predictive distribution, right? So maybe we don't actually care that much about interpretability, but if you give me you give me data that I train on, I want that my out of sample predictive distributions when I'm confronted with new data are good in the sense that they're well calibrated, that maybe the mean squared error is low. Whatever your notion of good is, we want good predictive distributions. So that's another desiderata that you might have. Another is that we can deal with high dimensional covariates, right? So methods that might work when you have 10 covariates might not work very well when you have a million covariates, right? And that's a different regime, and it may require special treatment. And finally, another thing that we would like is something that can deal with correlated covariates, right? And this sort of directly ties in with the third point, right? Because often when you have high dimensional covariates, you inevitably have covariates that are highly correlated. And you want a method that can deal with that gracefully. Okay, so let's just quickly review some of the most simple approaches to linear regression. So the classic method, of course, is that of ordinary least squares. So we choose a coefficient vector beta that is the minimum of this loss term, right? Which is literally just trying to find that beta, which forms a line that has the least squared error around that line with respect to the data y. And the nice thing about ordinary least squares is that there's a nice closed form solution for beta in terms of x and y. But one problem with this, of course, is that this solution is even well-defined if n is less than p. So if you have fewer data points than you have covariates, this object here isn't even defined. And so this sort of approach isn't even applicable in the high dimensional setting where p is generically larger than n. So a simple workaround to do something that looks like ordinary linear regression in the high dimensional case is to make sure that this X transpose X matrix here, which we take the inverse of and is not invertible in the high dimensional case, we can make it invertible by adding a small, a diagonal matrix that is scaled by some small positive number lambda. And so for any positive lambda, this matrix at the top that we're inverting will be invertible. And so this ridge regression estimate is well defined. This ridge regression estimate can also be understood as, a solution of a minimization problem. We again have an squared error term, but we also now have a regularization term of the form lambda beta squared, where this beta squared is just the Euclidean norm squared. And this of course has the property that, for example, as lambda gets very, very large, you're going to prefer beta solutions that are very close to zero, right? So as lambda gets larger, you're regularizing your solutions closer and closer to the origin. And in the opposite limit, the estimates are less regularized. And there are a lot of different ways you might interpret what's going on in this formula. One way to interpret this is that this ridge regression estimate is just a posterior mean of a Bayesian linear regression model in which the prior precision over the coefficients beta is given by lambda. So in other words, if you sort of assume that the prior variance of the coefficients, so in other words, if you assume that the sort of effect size of the coefficients is of order 1 over square root of lambda, then that is equivalent to what we're doing here, right? So if lambda is large, you assume a priori that the coefficients are small. Okay, so much for the most basic alternatives. Later on, I'll talk a bit more about other approaches like lasso, but with that sort of very brief introduction, let me actually talk about the thing that we're interested in this talk, which is Bayesian variable selection. So Bayesian variable selection is another approach to doing generalized linear regression in these kind of high dimensional settings. And conceptually, you can think of it as a model-based approach that's looking for parsimonious solutions, right? So here parsimonious means that, if I have, let's say, millions of covariates, maybe I want to find solutions that only make use of 10 or 20 or 30 of those covariates, and the coefficients for the other covariates are set equal to 0. And so we're looking for solutions that don't make use of the vast majority of covariates, but make use of only a small number. And this is in contrast to ridge regression, where generically speaking, for any lambda that you choose here, all the betas will be non-zero, right? So it's true that if you have a lot of data, many of the betas might be somewhat small, right? Like if a given covariate X is not correlated with the response Y, then that ridge regression estimate will tend to be small-ish. And the degree to which it's small-ish will depend on, among other things, the value of lambda. But none of these estimates will be 0, right? There'll be 10 to the minus 3, there'll be 10 to the minus 4. And so if you were sort of to look at your estimates, if P is very large and you look at a bunch of ridge regression estimates, typically you'll have maybe a few betas which are large-ish, and then you'll sort of have a tail into smaller and smaller betas. And if you're trying to interpret that data, it becomes a bit more difficult. It becomes a bit difficult to know, like, where's the cutoff between actual significant fits, like betas that actually are well supported by the data, and betas that maybe are just, are just picking up on some possibly spurious correlation. And so while ridge regression is very simple and elegant, it doesn't give us sparse solutions, and that that makes interpretability potentially harder, especially in this high dimensional setting. And so Bayesian variable selection is really focused on interpretability, among other things. And the way it goes about that is by really trying to find parsimonious solutions, again, that only use a small number of covariates. So that's sort of what's happening at high level. Operationally, the way this works is that Bayesian variable selection is formulated as a model selection problem, right? So we're comparing different models. And the notion of model that we're using here is connected to which covariates we include in the model and which covariates we exclude from the model, right? So you can imagine a model that only looks at, say, 7 covariates, and another model looks at 12 covariates. And so we have this large nested space of models that is adding and subtracting covariates. And Bayesian variable selection is all about searching that space and defining that sort of search problem as a model selection problem. So let's make that a little bit more concrete, right? So we can imagine models where there's only one covariate, right? So I've listed 3 such models to the top. We only have one coefficient in each of these models. Then there's a whole set of models with 2 covariates. There's a whole set of models with 3 covariates, Also not depicted here, there's a null model where we include 0 covariates, and we just try to explain the output variable y entirely with noise. And this continues through all capital P covariates, right? So we get 2 to the P total models. And so the space of models here is very large. On the left, I've sort of shown this in equations, but another convenient way to sort of encode or graphically think about what's going on is that we have capital P many covariates, and each of them is going to be turned on or off. So we can depict this graphically as these little vectors, these p-dimensional bit vectors, where each element of this vector is either 0 or 1. If the element is 0, then that covariate is not included in that particular model. If it's 1, then that covariate is included in that particular model. And graphically, we get these little vectors that are, let's say, light gray and dark gray, where dark gray here corresponds to including that particular covariate, right? So we want to sort of search this space of models, and that's the name of the game in Bayesian variable selection. There's one kind of obvious difficulty with this, which is that there are 2 to the power P models. And if P is relatively small, like 20 or 30, or maybe even 40, we could, it's feasible to explicitly enumerate over that, because those are large numbers, but not insanely large numbers. But 2 to the P very quickly becomes very large. So for example, the estimated number of atoms in the known universe, which is like in an entire like Hubble volume, this is like many, many, many light years of space. It's a very large, unfathomably large amount of space. Even in that amount of space, we have an estimated 10 to the power 80 atoms, and 2 to the P already exceeds that for 266, right? So if P is, say, 1,000, it's completely infeasible to somehow enumerate this space. 2 to the P is incredibly large, and so we need a different strategy. Okay, but before we get to strategies about how we might actually search this space, we need a criterion to tell us like which models do we prefer, right? Because somehow some models in this space are better and some are worse, and we want a sort of clever criterion that helps us choose amongst these different models. And as is commonly the case in various parts of statistical machine learning and elsewhere, the way we're going to do that is by somehow balancing model fit and model complexity. Right? We already did this in the case of linear regression. In the case of linear regression, when we did bridge regression, model fit was just the squared error, and model complexity had to do with the coefficient, the norm of the coefficient of beta. Right? So that was already an example of this paradigm. We're balancing model fit and model complexity. Bayesian variable selection is still going to be in that broader paradigm. The difference is that the notion of model fit and model complexity that we're going to be using is different. But at the end of the day, to each of these 2 to the power P models, we are going to effectively assign a score, where that score is a balance of fit and complexity, right? So high scoring models have good fit and low complexity. And so we'll see how that works in detail shortly. So the way this is done in this Bayesian variable selection setting is basically to formulate everything as a Bayesian model, right? So we're going to have this sort of meta model, which is going to help us choose amongst little models, where the little models are like the actual linear regression or logistic regression models, but we have this bigger model space that we're going to be using to actually do model selection. And so in that setting, the natural notion of model fit that is common in the Bayesian setting is the marginal probability of data given the model. So that's depicted here on the left, P of y given x and gamma. Recall that gamma here is, again, this p-dimensional vector of zeros and ones that specifies which covariates have been included and excluded in the model, right? So that's basically how we're parameterizing our space of models in this Bayesian variable selection setting. And P of y given x comma gamma is this marginal probability. And this is what you get when you integrate out all the variables that make up this model. And this is sort of the natural Bayesian notion of how well, given your prior assumptions and given your likelihood assumptions, how well that particular model fits the observed data well. And we're going to tie, along with that notion of fit, we have to introduce a notion of complexity. And the way that appears in this Bayesian setting is through a prior probability on the model, right? And this could in principle be just about anything as long as it's valid probability. But the general idea, right, is that we want the prior probability on models that include a small number of covariates to be larger than the prior probability of very complex models that have a very large number of covariates. That's like our basic fundamental assumption, is that this data should be able to be explained in a parsimonious manner using fewer covariates. And models that are very complex are a priori less likely. But the key word there, of course, is also a priori, right? So the data really says, you know what, I really need to use this very complex model to explain this data. It's impossible to get decent fit using, let's say, 4 covariates or 5 covariates. It will naturally, use more if necessary, but whenever possible, it will use the least complex model to fit the data at hand. Okay, so again, this Bayesian variable selection procedure is formulated in the vanilla Bayesian paradigm. And what that means is at the end of the day, we're going to get a posterior over models. Recall that we're parameterizing our models by gamma, this vector of zeros and ones. And so the thing that we would like to compute in this setting is the posterior over gamma given the observed data. And to compute this, all we would need to do, at least in principle, is compute this numerator, which we know how to do. It is relatively easy for most models, perhaps. But then there's a very difficult thing, which is that we would need to compute this denominator, where we sum over all 2 to the p models to turn this into normalized posterior distribution.
Martin, I think there was a question in the chat of how this, or is this a form of, I missed it actually. Thank you, feature selection.
Yeah, so variable selection is just another word for feature selection. Here, variable refers to which covariate we're using. Yeah, the one thing to keep in mind though is, often in feature selection methods, you just, you end up with, you choose one specific set of covariates. Whereas here, we're actually reasoning about this entire space of models. And if you want to, you can go at the end of the day and make a hard cutoff that says, I'm only keeping these features or these covariates. But you can also actually just reason about the entire space of models without making any explicit hard choices about throwing features in and out. The model, each and every one of these models makes a specific in and out hard choice, but we're reasoning and searching over the entire space. And so in that sense, it's a bit fuzzier than the most vanilla notion of feature selection.
Yeah, just a quick question about the interpreting the posterior. So if it's posterior over models which are have one of 2 n features, how do you interpret 2 models with the same posterior probability that have very different sets of features? Is there some kind of continuity imposed where it's different models which are close by, with respect to just one feature, have a similar?
Well, it depends what your notion of prior probability here is, right? But the simplest one would be the simplest, prior probabilities of our model parameterized by gamma is going to be something that depends just on the total number of covariates, right? So you could have more fine-grained notions if you have some prior information that you want to include. But a priori, if you believe your prior and if you believe your likelihood, then this is what the Bayesian posterior over model selection looks like. And if you have 2 models, let's say, that have similar posterior probabilities, then according to those beliefs, this is just telling you that those are kind of equally valid. And without making further assumptions or without further data, there's no reason to prefer one over the other. Does that answer your question?
Yeah, it does answer my question, but I still find it a bit unintuitive. What you do in that situation where you have these, like in the ridge regression example, right? You have each feature is ranked, so you could do something like take the top n and then.
Yeah, well, so we'll see later on sort of how you actually end up using these kinds of inferences. But typically, what you do not choose you do not end up choosing the model with highest posterior probability. The whole point of this, at least from my point of view, is to actually leverage the fact that you're searching an entire space of models. So at the end of the day, we're not going to often make a hard decision about saying that this is the particular best model. We can sort of use summary statistics that encapsulate what's going on in the posterior as a whole without necessarily, choosing one specific model explicitly. Although we can do that, but we needn't do that.
Great, thanks a lot. That was really helpful.
Okay, so just to sort of summarize in words, Bayesian variable selection is formulated as a model selection problem, and it's defined with respect to posterior distribution. And the idea is that the posterior mass will concentrate on the most parsimonious models that explain the data, right? So these models need to both be parsimonious so that the prior is, so they have high prior probability, but also they have to explain the data and get good model fit. And there's going to be a balance there, and that balance will be encoded in this posterior distribution. And that's the basic idea. Okay, so that was all pretty generic. So let's actually zoom in on a particular class of models that makes this a bit more explicit. So now let's look at linear regression in particular. Right, so before we just treated model as a black box, and we said model is parameterized by gamma. And that's, that encapsulates the main idea. But now for a specific model, we need to actually, make a few more assumptions, even though we're still in that generic paradigm. So let's look at a particular class of models for linear regression. So again, we're going to introduce a vector, a p-dimensional vector of gammas, where each element of gamma is drawn from a Bernoulli distribution. And the prior probability is governed by H, where H is a prior inclusion probability, right? So if you imagine that you have, let's say, 1,000 covariates, P is 1,000, and you make H equal to, let's say, 0.01, then that says that the expected number of a priori covariates that you would include in your model is equal to H times P, which in this case would be 10, right? So that isn't saying that we're only looking at models that have exactly 10 covariates, but that sort of is how you would interpret the sort of level of regularization that H is setting on our model space. Okay, so once we've drawn a set, now that, by making this Bernoulli assumption on gamma, we've effectively defined our prior probability over gamma, right? This is our complexity penalty. Models with many, many covariates turned on are penalized, and each of them is basically penalized by a factor of H, right? So if we have 100 covariates, we have to pay a factor of H to 100. And if H is small, that quickly becomes a very large number. And that's why models with very large numbers of covariates are strongly unpreferred by the prior. Okay, since this is a linear regression model, we're going to have a normal likelihood, which is depicted in the final line. That normal likelihood has a covariance, or a noise term, a sigma squared. And we're going to have to draw that sigma squared from some prior distribution. The vanilla choice in this setting is to use an inverse gamma distribution. A common choice is to, if you send the parameters of this inverse gamma distribution, namely mu naught and lambda naught, to 0, then that actually, this inverse gamma distribution becomes 1 over sigma squared. So that's sort of a common limit, to just say that the prior over sigma squared is an improper prior that's proportional to 1 over sigma squared. And that's a common choice. The actual details of that prior don't matter too much, because typically you have hundreds or thousands of data points, and so the sort of posterior over sigma is relatively narrow, because you kind of have enough data to pin down what the kind of level of unexplained noise is pretty easily. And then the important part on this slide appears in lines 3 and 4, right? So now we're going to draw coefficients beta. First, I'm going to draw a coefficient beta naught that's governed by some normal distribution. And this will be just an offset term or an intercept term. And then I define beta sub gamma by, again, drawing from another normal distribution. And the important point here is that beta has gamma as a subscript, right? Because gamma controls how many covariates we're actually, entering into our model. So if gamma is, for example, 1, 1, 0, 0, 0, and then lots of zeros, then beta of gamma is only two-dimensional, because we're only including 2 covariates. And so this beta of gamma will be a two-dimensional variable. And once we've drawn all those variables, then we can form a linear predictor in the normal way, beta plus x times beta. And that will define our actual predictor. And around that predictor, again, there's this sigma squared noise, and that will define our likelihood. So y is drawn from this normal likelihood. And the Bayesian variable selection is entering into here because we have gammas at the top, and we're basically if we just set gamma equal to one specific value, like 1, 1, 1, 1, 0, 0, 0, we would just have one and only one linear regression model. But because we also have a prior over gamma, we actually are sort of explicitly modeling a space of 2 to the p models. And so this is sort of, there's sort of a big meta model about which is the gammas. And then for each gamma, we have a little linear regression model, and we're going to sort of jointly do inference over this entire space.
You explain the noise term inside. If you can explain the noise for the two normal distribution for beta 0 and beta gamma, like.
Oh yeah, so the form of the prior over beta might look a bit unusual to you, but because it includes sigma squared, and sigma squared is nominal, is the noise that appears in the likelihood. But it turns out that if you make the variance of these, the prior over beta proportional to sigma squared, then it basically makes the math easier, and it makes it so that you can integrate out sigma. And so it's what's referred to in the Bayesian probabilistic modeling setting as conjugacy. So it's basically an assumption that makes the math much nicer, and it doesn't have that many consequences other than that. But then the point is, so sigma squared again is governed by this prior, but we also have we have these other hyperparameters here, which are tau, and those are hyperparameters that we can select to choose kind of the a priori effect size, right? So if we think that the betas are a priori very small, then we would, for example, use a very large tau, and that would say that we believe a priori that most effect sizes should be small. Now, again, priors can always be overwhelmed by the data, but our prior assumption will be controlled by, among other things, tau. So those are hyperparameters that you as the user have to choose. Yeah.
So I can't help but wonder, for a given data set with this Bernoulli prior on the gammas, aren't you effectively constraining to a space that's way smaller than 2p, because you would never, with any reasonable fit or any precision in your computer, you would sort of have a boundary that you couldn't cross in this space. You would never reach this 2p.
Oh yeah, we'll never look at the models with 500 covariates turned on.
Right. Okay.
But we don't know a priori, before we looked at the data, which ones are more and less likely to be turned on, right? So we still need to search this space. We don't need to search all of it.
Right, but given a certain data set, you can make an estimate of like how big of a circle you're searching in this space, or how big of a norm H you're kind of.
Well, effectively, remember, so if you take H and you multiply that by P, that's the expected number of a priori covariates. So that already kind of tells you the ring in which it's going to be focusing. But in practice, it might look quite a bit past that, just because maybe that's not enough to fit the data, right? So like if you set the prior number of covariates to 10, in your posterior, it might end up looking at models with let's say 30. It's not going to it's unlikely to look at models that have 500 or 7,000.
Right, okay.
But it will move past the prior if that's necessary to fit the data.
Okay, thank you.
So one of the nice consequences of this Bayesian variable selection approach is that because we're explicitly reasoning about including and excluding covariates, and because we explicitly introduced this random variable gamma, it makes it easy to compute something called the posterior inclusion probability, or PIP, which is a probability, or in other words, a score between 0 and 1 that we can compute for each covariate that basically encapsulates how much statistical evidence there is for the inclusion of that covariate in our model space if we want to fit the data, right? So if the PIP is, for example, very large, like 0.99, what that means is that in 99% of the models that are in the posterior, that covariate is included, right? So it's sort of needed to explain the data. Whereas if the PIP is very small, like let's say it's 10 to the minus 2, then that means that in 99% of the models that are in the kind of posterior region, then those models, 99% of them do not include that covariate, right? They are able to explain the data, at least to some fidelity, without using that covariate and by using other covariates. And so a covariate with a large PIP is one for which there's sort of large evidence that it's necessary to include that covariate to explain the data. And conversely, a small PIP covariate is one for which there's little statistical evidence for the inclusion of that particular covariate. And so this is useful because it's a nice interpretable score. It's between 0 and 1. We can use it to rank covariates and so on and so forth.
Martin, if you have like correlated covariates, and let's say you have covariates that are more independent, so then if we want to compare the BIP score among these cases, we need to keep this in mind, or there are other strategies to like.
Yeah, so there's definitely a dilution thing that can happen here, right? So if I have, let's say, if Y, the outcome, let's say we have two covariates X1 and X2, and they're highly correlated. And let's say each of them is independently able to explain Y very well, then what you're going to find is each of these covariates are going to have a pip of about 1/2, right? Because you could, once you've included one of them, there's no need to include the other covariate because it's effectively redundant with the other one. And so the posterior is going to concentrate on models that either have the first covariate and not the second, or it's going to concentrate on models that are conversely have the second covariate but not the first. And so you will end up with a pip of 1/2. So you definitely have this sort of dilution effect, and that's certainly something you need to pay attention to interpreting this kind of data. But I would argue that it's better than what a lot of other methods do, which is that they basically randomly choose one covariate, let's say the first covariate, and forget about the second covariate. And then you don't even know that there exists a second covariate, which was actually highly correlated with the first covariate. And there was also plausibly a good explainer of the data. And so this dilution kind of effect is potentially dangerous if you don't look for it or don't know about it. But it's, if the only assumptions you're making are linearity, and I want parsimonious models, and you're not bringing in any additional information about the covariates, then there's no reason why it should prefer one over the other. And so that's sort of the best you can do, and is arguably what you want.
With that, do you get sort of conditional pips where if you consider one of them to be positive, how often is the second covariate 0? Does that make sense?
Yeah, so I mean, in principle, if you were able to solve this inference problem exactly, or if you were able to draw samples from the posterior, then have these gamma vectors, right? So you could ask all kinds of conditional questions like, if gamma, if the third element of gamma is on, how often is the fourth element of gamma on, and so on. You can ask all those questions. Of course, as you start asking more detailed questions, it's going to be harder to actually do that computationally, but conceptually, you can do those sorts of things. Yeah, you can, ask questions like, oh, how, you might, if there's a covariate you're interested in, you might, and let's say it has a pip that's like 0.6, you might ask, oh, well, in those models where it's off, is there something else that turns to be on that's like sort of filling in that need? And you can certainly look for that sort of thing. Okay, so needless to say, this is a difficult inference problem. It's transdimensional because beta, actually the dimensionality of beta depends on gamma, right? So as we move around in the space, the dimension of the space is actually changing. So that makes it hard. It's also a mixed continuous discrete latent problem because we have both continuous and discrete latent variables. And as I've emphasized many times, model space is very large. So this is, a priori, this is very difficult. But in the last few years, people have come up with a few algorithms that are quite a bit better at doing this. And in particular, there's this cool algorithm from Giacomo Zanella and Gareth Roberts. And if you're a nerd who's interested in Markov chain Monte Carlo methods, I suggest you read this paper because it's very interesting. But the basic idea is that, we have this posterior that we're trying to explore, and it might be the case that there are modes that are separated, right? So for example, in the case we just talked about, where there are two covariates that are highly correlated, there's like a space of models over here where one is off and the second one is on, and vice versa. And these sort of modes in model space are actually separated. They're not like next to each other. And so it's sort of hard, it can be potentially hard to go from one mode to the other mode. And that's a regime in which these sort of MCMC algorithms often struggle. And so the basic idea of this inference algorithm is that it introduces an additional latent variable. So one of the tricks in MCMC algorithms is to make your problem easier, first you make it harder, where by making it harder, in this case, I mean increasing the dimensionality of the problem. And you can introduce an additional variable that controls a tempering or like flattening of the distribution. So you can imagine like flattening parts of the distribution. And by flattening it, it sort of becomes easier to move around in that direction. And so by sort of cleverly flattening an augmented space, you can sort of move around that augmented space, and then it becomes easier to do inference. You can sort of think about like introducing wormholes, and then you move through the wormholes and magic happens. But I suggest you read this paper if you want more details. Okay, so now that we've sort of discussed what Bayesian variable selection is and a little bit about inference, and this leads to the basic question of what can we do once we have these inferences, right? So we've done this inference problem, we have pips, we have estimates of coefficients and so on. What do we do next? So first of all, one very useful thing that you can do is just rank the hits by your pip, right? So now you have this interpretable score of the pip between 0 and 1. So you can use that to rank your hits, which is a sensible thing to do a priori. You can also do variable selection. So one thing you might do is, for example, only retain those covariates that have a pip larger than 1/2 and throw out all the other covariates. So you might imagine, a scenario that you might be in is like, let's say you are interested in drug resistance to some bacterium, and you have all this like bacterial GWAS data, and you figure out that maybe there's some small, you have some small number of like genomic variants that appear to be sort of causally linked to a resistance phenotype, and you can imagine that if you would like to use this actually in patients, say, like by actually looking at the particular variant that patient has of some bacterium, and it'd be easier if you could only look at presence or absence of a small number of genomic variants. And so in that case, you actually might want to do like strict feature selection, where you kind of limit yourself to some small number of covariates. That's sort of an example of a problem where you might actually want to do this kind of hard feature selection. Another thing that you can do is compute model average coefficients, right? So remember, this is a space of models, right? So in different models, the notion of beta is actually different, right? In the sense that like, if I have two models in which beta 2 is turned on, that beta 2 is of course linked to the same covariate x2, but it's also like, it's in a different, those models are different because there are other covariates that are sort of in the background that are being used or not used. And so that within any model, the estimates of beta are going to be different. And so we can sort of average over the space of models to get sort of a regularized estimate of beta, which is useful. We can also just compute model average predictive distributions, right? So for every model, there exists a predictive distribution. We also have a posterior over models. So we sort of take a posterior weighted set of predictive distributions across these different models. We get this sort of model average predictive distribution. And then also, to Ray's question earlier, we can look a bit more carefully at the details of the posterior, and like say, look at how gamma p is correlated with gamma q and so on and so forth, and try to like understand the correlation structure a bit in terms of like which models are sort of preferred versus not.
I have a quick question. So it's an approximate inference algorithm that you have to use with this prior. I guess, what are the limitations on what are the restrictions or assumptions on the posteriors that you actually get as an output from this algorithm? That is I mean, if you're using like variation inference, you'd know what the structure of your posterior look like.
Yeah, so in practice, like at least for certain likelihoods, where you're able to use an algorithm that's like this or similar to this, the inference algorithm actually works very well, right? So it's not scalable to like gigantic data sets. Like if you gave me a data set with 1 million data points and 100 million dimensions, like it would take too much compute time. However, if you're in a regime where it's actually computable, the inferences are actually quite good. It might take an hour on a GPU or 3 hours, or maybe it'll take 10 minutes on a laptop, and depending on the regime you're in. But it actually is, they're quite reliable. I mean, the bigger question is like, are you okay with the model assumptions? But actually the inferences are quite good once, if you're okay with the modeling assumptions. So there's a bit of theory behind this Bayesian variable selection stuff. I don't want to get into that too much, but like one theorem is these guys looked at sort of feature selection, and they sort of proved that at least under certain conditions, the median probability model is the single best predictive model. So here, median probability means take all the variables that have a pip greater than 1/2. So that maybe sounded like it was a more or less arbitrary thing to do, but it actually turns out that there are some like theoretical reasons to suggest that doing that or something like that, basically using a pip threshold, is sort of a coherent thing to do. So that's worth mentioning. We were talking a little bit about what you do with inferences. So to make that a bit more concrete, here's what the inferences might look like once you've actually run this on a data set. So here's a data set. Each of the first row is the name of some feature. The second column is the pip. And here I've ranked the different hits by pip. But then you also get other information, like you get say a posterior mean over a given coefficient, sort of averaged over all models. You get standard deviations and so on. So this is the sort of information you would get if you were to run this kind of method on your data. Okay, so in the remaining time, I just wanted to briefly discuss some sort of other competing methods for doing high dimensional statistics and sort of, penalized regression in the high dimensional setting. So one that's, of course, very popular and common is lasso. So this is, again, this sort of penalized regression problem where we're again, trying to minimize the squared error, but we also now have a penalty term. But instead of using the Euclidean norm, we use this one norm. And the consequence of using this different norm is that the lasso solutions tend to be sparse, right? So like most of the betas in this beta hat are equal to 0. Only some smallish number of them will be non-zero depending on the strength of your regularizer lambda. And so this lasso has a few nice features. It's conceptually simple. It's controlled by a simple hyperparameter, in this case lambda. It's relatively scalable, and it has a well-developed theory. So a lot of people use it for these reasons. It also has some less desirable features. So it has one sort of bizarre feature that it has is that if for n less than p, the lasso selects at most n variables, which maybe in practical terms doesn't mean a whole lot, because if you have hundreds of variables, if n is a few hundred, you probably don't want it to choose more than a few hundred variables anyway, but it kind of hints to the fact that there's some maybe weirdness going on here. The other thing is that it tends to deal poorly with highly correlated variables. In particular, it tends to do the thing that I described earlier, which is it kind of drops out certain variables, right? So if two variables are highly correlated, it's typically what it does is it selects one of them and gives it a non-zero beta, but assigns the other covariate a 0 beta, right? And so it just sort of picks one. If you have a group of highly correlated variables, it'll tend to pick one in the group, and it doesn't tell you or give you any sort of additional information to like figure out that like actually you're in some sense dropping information by doing so. It also turns out that it tends to be hard to get well-calibrated p-values and can control false discovery rates for the lasso. So that's problematic. So one sort of response to the lasso is something called the elastic net, which looks very similar, except this we add an additional regularizer here. So we have both a regularizer that's controlled by a one norm, but also another regularization term that's controlled by a Euclidean norm. And it turns out that this works better with highly correlated variables. In particular, if you do now have a group of variables that are highly correlated, elastic net is more likely to give, to keep all of them as opposed to just randomly keeping one and throwing the rest out. One of the cons, though, is that you now have two hyperparameters to deal with. Typically, these hyperparameters are chosen using some sort of cross-validation. So that becomes now a bit more expensive because you have to search over some grid of two hyperparameters. And it still retains some of the other problems with the lasso, like difficulty of computing p-values, for example. So now let me quickly discuss two Bayesian approaches. One is called the spike and slab. And so here we're going to put a prior over the coefficients beta that is a mixture of two normal distributions. One is a large variance slab and one is a low variance spike. And in a lot of ways, if you look at this model, it looks pretty similar to Bayesian variable selection. We're introducing capital P many Bernoulli latent variables Z, which are in very many ways similar to the gammas I introduced in Bayesian variable selection. But here, the key difference is that beta doesn't depend on gamma, right? So in the Bayesian variable selection problem, I'm treating each of those models as separate. Whereas here, there's only one beta, say one beta 2, one beta 3, and so on. And there's this prior, which concentrates a lot of the prior probability mass near 0. But it's nevertheless the case that we're not doing model selection. We're just using a particular prior over beta in one, there's one particular model, and in that model, there's a particular prior over beta, and that prior over beta has this form, but we're not actually comparing discrete models with one another. So it's very similar, but it's subtly different. Finally, another popular class of Bayesian methods for dealing with high dimensional regression is to use continuous shrinkage priors. So I should say one of the difficulties with this spike and slab approach is that it turns out it can be pretty difficult to do inference well here. Although there's recent work that has made some progress on this. Another approach is to, instead of using a spike and slab prior, is to use something that looks a lot like a spike and slab prior. And so here the idea is to just place a prior on the coefficients beta that concentrates near zero. And so we're in effect mimicking the spike and slab by a single continuous density. And so this will give us shrinkage, but it won't give us strict sparsity, right? Because there's still always non-zero probability for beta to be in the neighborhood of zero. And so we'll end up with inferences where a lot of the betas are small, but they're not strictly zero. And one particular sort of implementation of this idea goes under the name of the horseshoe prior, and it is depicted here, right? So let's just look at this plot quickly. So first of all, in blue, we have a student t distribution, which is a fat version of a normal distribution. So you see it has fat tails. And then we have a Laplacian distribution in red, which again is a fat tail distribution. But then in black, we have the horseshoe. And you can see the horseshoe actually has a singularity at zero, right? So these other distributions have, finite density, like a bounded density near zero, whereas this horseshoe prior actually has unbounded density near the origin. So it really is placing a lot of posterior mass near the origin. But in other respects, it's similar to these other distributions in the sense that it's fat-tailed, but it really sort of strongly prefers putting betas near zero. And one of the nice things about this is there are some pretty good inference algorithms that accommodate this. And in particular, if you're in a sort of moderate regime where you, let's say, only have a few hundred covariates, then it turns out that some black box inference algorithms like Hamiltonian Monte Carlo actually are applicable in this case. And that's nice because that means that, for a lot of these other models, like you really need to take advantage of conjugacy or some other like math tricks to make inference work. And that limits the class of models you can look at because only certain likelihoods sort of allow for those math tricks. But if you use a horseshoe prior, and again, you're in like a moderate number of dimensions, then it turns out that you can often use just about any likelihood you want, because then you can use a black box inference algorithm to do inference, and it will work reasonably well. It won't work well in a million dimensions, but it'll work pretty well in, say, 500 dimensions. So that's a nice benefit of that approach. Okay, so let me just quickly summarize. I tried to argue that Bayesian variable selection is a powerful method for doing inference in the high dimensional case of generalized linear regression. It has a number of nice properties. First and foremost, I think, is that the fact that it gives you posterior inclusion probabilities or PIPs, which you can use to rank your hits or otherwise reason about what your data is telling you. Also, recent algorithmic advances, but also just the fact that we have GPUs and better hardware means that these sorts of approaches are now scalable to larger problem classes. In very rough terms, maybe we can scale to tens of thousands of data points and 1 million covariates, sort of depending on details. But that wasn't the case, say, 10 years ago. So in that sense, this is for many years, this Bayesian variable selection was a bit academic because it just wasn't nearly scalable enough, and there just wasn't good inference algorithms. But I would argue that now we've entered, as of a few years ago, it's actually practical to apply this to at least certain classes of problems. You can't, maybe there might be data sets where you would like to do this, but they're still too big, but it's actually now become, I think, a useful tool. And so if you want to try Bayesian variable selection on your data, check out this open source package that I wrote that does Bayesian variable selection called Millipede. You can follow the link here at the bottom, and it supports a few likelihoods, including a normal likelihood for vanilla regression, but it also supports a few count-based likelihoods in case you want to do logistic regression or binomial regression or so on.
Why did you call it millipede?
Because a millipede has 1,000 little legs, and I guess you want to choose the right legs or something like that. Yeah. Also, so here are a bunch of references that kind of are relevant to some of the things I talked about, but also please check out some of the previous MIA talks that are also about high dimensional regression or high dimensional statistics sort of writ large. Especially, one thing that I didn't talk about at all that's seen a lot of interest in the last few years is something called knockoffs. And so there's some nice talks about that. So make sure to check that out if you're interested. Okay, thanks for having me. So as Ray said, I'll now talk about some applications of Bayesian variable selection. And so the outline is as follows. First of all, I'll very briefly review what Bayesian variable selection is for those just joining the seminar. And then I'll talk about two applications. And then finally, I'll give a very short demo for a piece of software that I developed that helps you do Bayesian variable selection. Okay, so what is the context? The context is that of generalized linear regression. We have a data set with, let's say, n data points. We have covariates x. Each x is a p-dimensional vector in R to the p. And we're doing generalized linear regression in the sense that, we combine some linear combination of covariates x with coefficients beta. We send that through some link function f, and that allows us to predict the response variable y. If the response variable y is, let's say, logistic regression, then this f might be, a logistic function, but the simplest case, f is just the identity function, y is continuous, and we're just doing vanilla linear regression. But the point is that the setting of interest here is that of linear regression with capital P many covariates. And so Bayesian variable selection is a model-based approach for identifying parsimonious explanations for the observed data y given the covariates x. And one that I will argue is especially useful in the high dimensional setting where you have lots of covariates. And so here parsimonious just means that we're trying to use as few of the covariates as possible to explain the output or response y. And sort of the way this works is that we're going to explicitly reason about including and excluding different covariates, right? So you can imagine this large space of models, including models where there's only one covariate turned on, models where there are two covariates turned on, models where there are three covariates on, and so on and so forth. And we're going to sort of explicitly reason about this model space and try to find parsimonious models that only use a small number of covariates to explain the response y. And this ends up being formulated as a model selection problem, and we get a posterior distribution in the typical Bayesian manner, and the posterior mass ends up concentrating on the most parsimonious models that also explain the data. And one of the nice side benefits of kind of explicitly reasoning about inclusion and exclusion of covariates is that it makes it easy for us to compute a posterior inclusion probability, or PIP. And this is basically a probability or a score between 0 and 1 that kind of encapsulates the statistical evidence for the sort of connection between the pth covariate and the response y, right? So basically this is saying if the PIP is large, like let's say the PIP is 1, this is saying that in the posterior, the vast majority of models that fit the data well actually needed to use this covariate to explain the data. So the fact that we get this PIP is a very useful aspect of Bayesian variable selection. Okay, so with that very short recap, let me actually talk about most of the talk. I'm going to talk about two particular applications of Bayesian variable selection, which will also hopefully make kind of this set of ideas a bit crisper, at least in terms of like how you might actually go about using this. So the context for the first portion of this talk is the fact that we have sequenced a very large number of SARS-CoV-2 genomes. As of a few days ago, we're above 13 million sequences, right? So we've never sequenced virus like this many times, right? There's no other comparable data set. We've never seen a virus sort of evolve in real time the way we have here with SARS-CoV-2. So this is sort of a very large and rich data set. And as a large and rich data set, it might motivate us to ask some more or less ambitious, detailed questions about what's going on. These questions might include, for example, which mutations are driving the pandemic, which variants are the most fit, which newly emergent variants should we be paying attention to, and so on and so forth. So before trying to formulate a model that might attempt to answer some of these questions, let me familiarize you quickly with some of the features of this genomic surveillance data. So here's a plot taken from NextStrain that shows sort of the dynamics of variant frequencies in SARS-CoV-2 over the last 6 months. And in particular, what we see here plotted is the variant frequency for several variants, different Omicron subtypes. And you can see that, first of all, the variant frequencies are, of course, varying across time, but there's also this sort of prototypical pattern where, one variant sort of goes up and then it goes down because it's sort of overtaken by some more recent fitter variant. And so if any model that we're going to write down needs to capture these kind of replacement dynamics. The other thing is that in this, this plot here, this is sort of from global data. And so these sort of variant frequency curves are quite smooth. But if we start zooming in on the data, we see that the data is actually pretty stochastic, right? So here we see a very similar set of time series curves. Here we're showing variant fractions for one particular variant, B117, which was also known as the alpha variant. And each of the time series curves here corresponds to a different region. Time is across the horizontal axis. And what you can see is that, while there's sort of broadly similar behavior between different regions, for the most part, in the sense that, at the end of 2020, beginning of 2021, the variant frequency went up, and then it plateaued, and then it went back down. Between regions, there's actually quite a bit of variability, right? So like, this variant hit the UK early, but, hit Japan quite a bit later. It never even really took off in Brazil. But also, even within any region, there's quite a bit of stochasticity from time bin to time bin, right? So although, large amounts of money have been spent to sequence SARS-CoV-2 genomes, we're still only sequencing, a small portion of the actual, patients that end up getting SARS-CoV-2. Even in the UK, which had probably the most ambitious surveillance program, where they were sequencing maybe something like 1% of genomes, right? And so we're not seeing all of the data, and so there's quite a bit of stochasticity in this data. And so any model that we write down needs to sort of explicitly reason about this stochasticity if we're going to get reliable inferences. And so these sort of considerations led us to write down a model called Bayesian viral allele selection, or BVS. And at high level, this model kind of consists of 3 components. The first is that of data representation. So the data representation that we're going to be using in constructing this model is that of allele frequencies. And we'll be looking at well-sampled regions. And these allele frequencies will be for some large number of non-synonymous amino acid substitutions that are observed in these genomic samples. You could in principle use some different representation, like you could do a nucleotide level representation, but we'll be using an amino acid level representation. And one key feature here is that these are frequencies, right? So frequencies are between 0 and 1. We're not trying to model the sort of absolute growth of SARS-CoV-2 genomes over time. We're really asking questions about frequencies and relevant prevalence. And so this sort of simplifies things, because absolute behavior is determined by things like lockdowns and other human behavior, whereas sort of relevant or sort of relative frequencies are kind of more directly linked to the relative fitness of different variants. And that's precisely the sort of information that we're interested in extracting. Second of all, in terms of modeling, we're going to be specifying a diffusion-based model. And so that sort of diffusion is going to sort of take into account this stochastic element that I referred to on the previous slide. And then finally, in terms of sort of our high-level statistical assumptions, we're going to be inferring selection effects under the assumption that most alleles are neutral, right? So we observe thousands and thousands of different alleles, different mutations in these SARS-CoV-2 genomes, but we're going to assume a priori that most of these alleles are neutral, or at least nearly neutral. And so this is just needed in some sense for statistical reasons, right? Like we have this very high dimensional hypothesis space. There are many alleles. And so doing something like this will help us regularize our estimates, but also it's sort of biologically plausible that most of these alleles probably are neutral or nearly neutral with respect to viral fitness. Okay, so let me just quickly remind you what diffusion is. So, diffusion is something you're familiar with from daily life. For example, whenever you make a hot cup of tea. Here we have a simple experiment where someone is dropping colored dye into a beaker of water. And there are just 1 or 2 features I'd like to highlight here. So first of all, we have this sort of expanding cloud of dye. But if you sort of look at the details, it's sort of quite noisy, and these wisps are sort of different in different directions. And you can imagine if you redid this exact same experiment under the exact same conditions, the cloud of dye would still look different from sort of experiment to experiment. There's just sort of an inherent level of stochasticity here. But also on top of the inherent stochasticity, there's also sort of a deterministic component. And in this case, that deterministic component is provided by gravity, right? Gravity points down. And so even if I repeat this experiment 100 times, this cloud of dye is moving down. We'll see that sort of in each and every one of these experiments, right? So the nice thing about diffusion is it sort of brings together both a deterministic component, but also a stochastic component, which is precisely the sort of behavior that we observed in these allele frequency curves, right? Like all, allele frequencies tend to be going up, let's say, for a given allele in a given time period, but there's also still quite a bit of noise around that. And that's exactly the kind of behavior we need to account for if we're going to model this surveillance data. So I wanted to point out that there's a really neat paper earlier this year by these folks that take some ideas from population genetics, in particular this sort of diffusion-based idea, and applied it to SARS-CoV data. And so basically what I'll be doing in the following is taking some of those same ideas, but combining them with Bayesian variable selection as a way of sort of doing the inference in a way that's that's hopefully more robust and sort of statistically efficient. Okay, so let me just quickly at high level detail how this diffusion-based thing works. So the basic sort of modeling assumption that we're going to make is that individuals are infected at a given time step, and individuals cause other people to be infected in the next time step. And that infection process will be governed by some distribution, in this case, a negative binomial distribution, right? So there's a sort of an inherent level of noise. One person might infect 2 people, another person might infect 4 people, and that inherent noise is something we want to explicitly take into account. Also, we're going to assume that there's a basic reproduction number here denoted by R. So if R is 3, then R is the mean number of individuals I expect for an infected individual to infect at the next time step. So that's sort of the basic epidemiological model that we'll be using. But so far I've only talked about variants, or I could also refer to them as lineages. But to really make this interesting, we want to connect the variants back down to the genotype, right? We're really interested in what's happening at the allele level. And to do that, we need to make additional assumptions. In particular, we need to introduce some sort of fitness model that connects the basic reproduction number R to the actual alleles and mutations that we observe. And we're gonna make the simplest possible assumption, which is that of a linear fitness model. And so we're gonna introduce selection coefficients beta, capital A, many of them. In the following, A will be about 3,000, right? So we have about 3,000 selection coefficients for each of the 3,000 alleles that we observe in our surveillance data. And the genotype of each variant, say, any particular variant only has some small number of these different mutations, and we're going to assume that its fitness is a linear combination of a small number of those selection coefficients that correspond to those particular mutations. So this is just a basic linear fitness model, and this is what allows us to connect the genotype to the basic reproduction number. So before we go on, sort of zooming out, it might be useful to make a sort of hand-wavy analogy, and that is the following. So imagine that we observe a short video of diffusion in 100 glasses of water on a 3-dimensional planet, and our goal is to infer the direction of gravity, right? So this is, what's going on here. So these 100 glasses of water are going to be the 100 regions in which we observe, these genomic samples, and the 3,000 dimensions of this planet are the 3,000 alleles that are observed. And the direction of gravity is this sort of deterministic component in these diffusion dynamics that, in our case, is going to correspond to selection effects, in particular the fact that certain variants are more fit than other variants, right? And so if you imagine sort of simulating the pandemic again and again and again, the sort of details of some of the are going to look different from pandemic to pandemic, but certain things are going to, certain, like, let's say, alleles that actually have a causal effect on increasing fitness, you'll expect to see those to go up in all those pandemics. And that's the sort of, it's identifying those alleles that are sort of causally linked to increased or viral fitness that we'd like to identify. And given this analogy, you can imagine why this might be statistically hard, right? If you have this sort of noisy videos of these clouds expanding in 3,000 dimensions, you can easily make a mistake and pick the wrong direction And so we need to we want to use some tools from statistics to regularize our inferences in this sort of high dimensional setting. And so given the primer, it's no surprise that we're going to be using ideas from Bayesian variable selection, right? So we're going to assume that most of our selection coefficients beta are strictly 0, or sort of in more biological terms, we're going to be balancing data fit, right? We're trying to fit this surveillance data, but we're going to balance that with our neutrality assumption, which is to say that we're going to prefer parsimonious solutions over complex solutions, and we're going to assume that most alleles are neutral or nearly neutral. And so the way this model ends up being formulated is, at least at a high level, pretty simple. We have a capital A dimensional vector of Bernoulli latent variables, where each of these Bernoulli latent variables explicitly controls whether or not a given allele is neutral or not. If gamma A is 1 for a particular allele, then that means that it's not neutral. And so we will include it in our model, and we will draw a selection coefficient for that particular allele. And then that selection coefficient will end up in our likelihood. I don't really have time to go into the details of how this diffusion-based likelihood works, but an important feature of it is that it's actually a normal distribution, a multivariate normal distribution, and these beta coefficients enter into a deterministic drift, which here is depicted by D, and it enters into D linearly, so that makes inference relatively easy. And the point is that we want to find those betas that give us a deterministic drift component of this diffusion that both accounts for the fluctuations we see in allele frequencies in our data, but that also is parsimonious and takes our neutrality assumption explicitly into account. It's also worth pointing out that there's some very clever wordplay here, where selection is both used in the genetic sense of genetic selection, but also in the sense of Bayesian variable selection. And so that's why this method had to be given the name that it was given.
Just a quick question. So in this data, you have like an explicit linear dependency between some of the covariates, right? Because if you have one amino acid,
sure. I mean, this is a sort of autoregressive model in some sense, but all these, what we're effectively doing is modeling. The outcome here is really a difference in allele frequency changes. And the thing we're using allele frequencies at the current time step to model the differential change in allele frequency in the next time step. But if you squint, this actually has the same math as a linear regression problem, more or less. And so it is a bit strange, but the point is that this is actually sort of a well-formulated likelihood that gives us the takes into account the kind of behavior we'd expect from allele frequency.
Yeah, no, I kind of meant from the selection coefficient, if you have, maybe I'll get back to you.
Are you asking if it's sort of a conditional selective pressure on certain alleles? Like it's only happens if there's another allele that.
Oh, no. I'm saying that if you have one allele, then you automatically don't. If you have one amino acid substitution at a position, then you automatically don't have that substitution. You don't have another one, right? So like if you have an A at a position, you don't have an M or whatever.
Yeah, well, so it turns out that that really doesn't matter to first order. So this model can be both, it can be formulated naturally in variant frequency space, and then you can sort of shift to allele frequency space, and then it actually doesn't matter. Well, so first of all, there aren't typically that many, there aren't multiple alleles at one site. There's usually just one residue, one common substitution. There are a few cases where there might be multiple ones, but that's in any case, not that common. But it also just doesn't end up mattering in terms of the math. Yeah. It does matter in the sense that, if you want, let's say you have a substitution, two substitutions at the same site. If you wanted to assume that they have the same effect, or that their effects should be similar, then maybe you would need to put in additional assumptions, but we make no such assumptions. Okay, so to sort of recapitulate, we're going to be searching over this space of 2 to the capital A models, where A is the total number of alleles. We're going to be using some Markov chain Monte Carlo method to do so. And we're going to be preferring models where only a small number of alleles are non-neutral, and most of them are neutral. And again, we're using this Bayesian variable selection approach. So one nice thing that we'll get at the end of the day is we'll get PIPs, posterior inclusion probabilities, that will help us sort of rank the alleles. and help us identify which alleles are likely actually, feeding into differential fitness in SARS-CoV-2. So before I get to actual results, let me just quickly show some results from some simulations. So I don't want to get into what's going on here in detail, but like basically along the horizontal axis, we have the number of alleles, right? So I'm simulating a bunch of pandemics with a given number of alleles. In every pandemic, there are only 10 causal alleles. So there are 10 alleles that actually have a causal effect on differential fitness. And plotted on the vertical axis is some measure of success, right? Basically, we want, if possible, we want this measure of success to be 1. If it's 1, that basically means that we found the 10 causal alleles and didn't make any mistakes. And there are basically, there are a few things we see here. So first of all, as you increase the number of alleles, it gets harder to do this, which is not surprising because your hypothesis space is increasing dramatically. Also, as we go from the left plot to the rightmost plot, we're increasing the number of regions. So we basically have more data. We're observing sort of more mini pandemics. And so of course, it's easier to do a better job when you have more data because you're less likely to be fooled by fluctuations because you sort of can compare what's happening in different regions to different regions and sort of extract the commonalities more easily. And then finally, our approach, which is plotted in blue, seems to do the best across the board compared to these other approaches I compared against. So this at least gives us some belief in the fact that if we run this kind of method on actual genomic surveillance data and rank hits by PIP, which is how I ranked hits here, that we should do a pretty good job of being enriched with alleles which are actually causally linked to the differential fitness. Okay, so let me actually just then jump into the results. So we downloaded 6.9 million sequences from 128 regions using data through April 18th of this year. We used 2,975 non-synonymous amino acid substitutions. And recall that our model is going to deliver 2 kinds of inferences, right? So first of all, we have allele level inferences because we have these betas. We also have this linear fitness model that connects beta to the basic reproduction number. And so we also are going to get variant or lineage level inferences. And so I'm going to first start with the lineage level inferences. And so what I've done here is I pulled out a few sort of well-known SARS-CoV-2 lineages. And in the rightmost column, we have the sort of estimated growth rate or the estimated relative reproduction number relative to the original Wuhan A variant. And these are ranked from top to bottom from sort of highest fitness to lowest fitness. And you can see here that this ranking sort of recapitulates the observed replacement dynamics, right? So this is really more like a sanity check, but it shows that, alpha and beta have a lower fitness than delta, which in turn has a lower fitness in Omicron. So this is at least a good sanity check and sort of recapitulates what we would have expected to see. Here at the time, remember this is using data through April 18th. These are the top 20 lineages as ranked by this model, right? So we're using this so-called Pango notion of lineages. There are a few thousand Pango lineages. This is a pretty fine-grained notion of SARS-CoV-2 lineage. And you can see that there's, quite a lot of lineages that are sort of towards the top of, are estimated to have large fitness. And these are all basically different subvariants of Omicron. And if we were to run the model today, we would also see a bunch of subvariants of Omicron because Omicron continues to sort of dominate the dynamics of what's going on. Okay, so now let's turn to allele level inferences. These are perhaps more interesting because they're sort of less immediately obvious from the data. So what I'm showing here are the inferred selection coefficients beta that we infer. So recall that we have about 3,000 alleles. So each of these red dots corresponds to one of these 3,000 alleles. Plotted on the vertical axis is the inferred magnitude of these selection effects. So in practice, almost all the selection effects that we're able to identify are positive, right? So there isn't too much, there are a few alleles that appear to have negative selection effects, but for the most part, we primarily see evidence of positive selection. And also the size of these dots corresponds to the pips. And so first of all, we see that most of these dots are at the very bottom, like near 0, right? Which is in accord with our prior assumption, but it is also something that, you can sort of see in the data, right? Like you can explain the observed surveillance data without having to turn on selection effects at hundreds and hundreds of alleles. in practice, you can get a pretty good fit by, let's say, assigning non-zero betas to 100 alleles or thereabouts. And you can also see that, so in this plot or along the horizontal axis, we sort of have the genome splayed out linearly, and the genome has been demarcated into different genes. In particular, we have the spike gene or the S gene demarcated. And you can see that a lot of these hits cluster in a few specific regions, including, of course, the spike gene, but also a few other genes like the nucleocapsid where you would, you might expect to see hits. So there are no, a lot of that is sort of what we would expect, but there are also some hits that we observe in regions of the genome which are sort of less well characterized. And it's not entirely clear what's going on there, but the nice thing about this kind of analysis is that it's, it's genome wide and it's agnostic, right? We didn't assume a priori that all the action is in the spike gene. We want to just look at the entire genome. Okay, so now let's look at the top hits. So here are the top 10 hits we saw. A lot of these are in the spike gene. A number of these mutations are sort of well known and well characterized by this point. So for example, this, I believe this EEC, the so-called EEC mutation, the third one here, is implicated in immune escape. And a number of these others are also sort of, well known to epidemiologists. So there are no huge surprises here. And, as you can see here, these are all like very high PIP estimates. If we keep going down, of course, the PIP starts decreasing, but there's something like, I don't know what the exact number is, but let's say there are 50 hits that have a PIP above of 1/2 or something, right? And we start seeing decreasing PIPs, but the PIP turns out to be a useful way to rank some of these hits. So far I've just shown you tables and plots, but another cool way you can try to visualize some of these hits is graphically like this. So what do we see here? So in yellow and blue, we see the S1 and S2 subunits of this spike protein. And in magenta, we have the ACE2 receptor, which is sort of the main entry point for spike that, that SARS-CoV-2 uses to enter epithelial lung cells. And the hits, some of the top hits in our analysis are denoted in red. And on the right, we're just zooming in. And so what you see is that actually a lot of the hits appear in this interface region between ACE2 and the RBD region of spike. So there are no surprises there, but it's nice to see that this sort of agnostic genome-wide analysis sort of is able to pick out some of this sort of plausible behavior that we're seeing here. Okay, so just to quickly summarize, I introduced this method called BVS, or Bayesian viral allele selection. Basically, at high level, we combined a diffusion-based likelihood with Bayesian variable selection. And I asked some questions at the beginning of the talk. And I just showed you some lineage level and allele level inference results. And I would claim that at least for some of these first few questions, we're able to get pretty good answers to some of these questions. There's some more ambitious questions we might answer. For example, we might try to ask questions about vaccination dependence or epistasis. And I've also done some analyses to that effect. These are inherently harder questions to answer, so it's a bit harder to evaluate. But if you're interested in that or other sort of sub-analyses that we did, of which we did very many, please check out the preprint. Okay, so now I'm going to move on to the second and final application. So switching gears entirely. So I'm going to talk a bit about some CRISPR tiling screens. And so some of my collaborators are developing experimental techniques to do CRISPR mini tiling screens. And so let me first discuss the motivation before I get to the actual analysis portion. So this motivation will be familiar to many of you, but the generic idea is that genetic variation is associated with many human diseases. It turns out that a lot of GWAS hits end up being in non-coding parts of the genome. And so, this has led people to believe, among other things, that non-coding regions of the genome are probably playing a large role in gene regulation. And this is probably, implicated in various types of human disease. And so this leads to the natural question, which is how do we interrogate and characterize the function of putative regulatory elements, right? So if I have some hypothesis that there might be a regulatory element in a given region of the genome, how do I look into that more carefully? How do I validate that? How do I understand in detail what's going on? So there are different answers for how you might go about doing that experimentally, but one possible answer is to do some sort of CRISPR tiling screen where you go in and sort of perturb that region and try to understand which perturbations are linked to the actual target gene of interest, or actually have some causal effect on whatever the target gene of interest is. So let me describe a schematic experiment. You can imagine different experiments that differ in detail from what I'm about to describe, but this is just sort of a high-level cartoon description. So imagine that we identify some regulatory element that we're interested in that we think is linked to some target gene of interest. Maybe we identified it using ATAC-seq data or some other source of outside information. So then we prepare some large cell line, and we're going to transduce cells with CRISPR single guide RNAs that target this particular genomic region of interest. And so once these sgRNAs are integrated into these cells, some proportion of these cells will experience genetic editing in the region of interest, and so they'll be perturbed. And that will allow us sort of a window into what's going on, hopefully. And so to connect those, ultimately we want to connect genotype to phenotype, just like we did in the previous application, right? So this is a sort of a high level, we're doing the same thing. But in this case, the phenotype that we might get by, for example, sorting the cells, right? So imagine that we stain the cells, or maybe the cells have some, there's some cell marker that's related to the gene of interest, something that allows us to sort the cells. And in the simplest case, we sort the cells into let's say a high and low population, where low is linked to, let's say, low gene expression for the target gene, and high is linked to high expression for the target gene of interest. So now we have two populations of cells, and we're going to send both of them through the sequencer. So this is where this kind of family of tiling screens differs from some of the other many tiling screens that have appeared in the literature, in the sense that we're going to actually sequence these cells. And so we're going to see the specific alleles, right? We're going to actually see the edits. We're not going to just assume that a given, single guide RNA produced a specific set of edits. We're actually going to go in and look at what the edits are by actually sequencing each of these genomic regions. And so at the end of the day, once we've done that experiment and analyzed and sort of, assembled our genomes and so on and so forth, we end up with a data representation that might look something like this. So we're going to observe n distinct alleles in this sort of target region. N might be a few hundred, it might be a few thousand. It depends, on the details of our experiment. And because we sequence these two populations of the cells, the low and the high, we're going to get read counts that support, each and every one of these alleles. And the sort of number of reads that come from the high population will, generically differ, of course, from the number of reads that we get from the low population. And so this gives us some weak signal as to, whether or not that allele is linked to high or low gene expression, right? And so this data is going to be pretty noisy because we're looking at, reads, and there's a number of different sources of noise that we might worry about. But at the end of the day, we get a bunch of alleles, and each of these alleles is going to be associated with a high and a low read count. And we can imagine encoding the alleles using some sort of p-dimensional one-hot encoding. Which encoding we use is going to depend on the nature of the edits that we saw. So for example, we use Cas9 and we use indels, then we might use one encoding. We use base editors, then we might use a different encoding to account for the fact that we're actually introducing different substitutions at at the same site. So the details of encoding are going to depend, but at the end of the day, we've sort of set this up as a generalized linear regression problem, right? We have this p-dimensional set of covariates that corresponds to this one-hot encoding. We have these outcome variables H and L, and so one way or the other, we can turn this into a generalized linear regression problem. And how we do so is, is going to depend a bit on which assumptions we want to make about the data and how noisy the data is. You can imagine doing different things. Here are two examples, right? So we have to choose a likelihood, or in other words, choose a link function in the generalized linear model. So one thing that we can do is we can take the difference of H and L, the high and low read counts, and divide by H plus L. And so this would be some continuous number between -1 and 1 that sort of encodes the kind of differential preference for H versus L for that particular allele. And we could just then go and apply a normal, assume that there's normal noise on top of that. You could also use some count-based likelihood, like a binomial likelihood. So there are different options here. But the point is, at the end of the day, we've set our data representation up so that this is a generalized linear regression model. And once we've done that, we can do Bayesian variable selection. And the reason Bayesian variable selection might be attractive in this setting is that a priori, we're looking at this relatively large genomic region. Maybe it's 400 nucleotides in length. And we believe a priori that probably most edits in that region have no causal effect. They have no, they're not going to change the gene expression level. There's only some relatively small portion, or maybe some small region in that larger region that actually is causally linked. That's where the sort of the regulatory element is sort of localized to some smaller region. And so we assume a priori that most of these coefficients, most of these edits actually shouldn't have any effect on our H and L counts. And so that's why doing this Bayesian variable selection thing is useful, both because it gives us a way of making a sparsity assumption, which is to say that we expect that most sites are not important, but also at the end of the day, it'll give us PIPs, these posterior inclusion probabilities, and that'll be just a useful way of helping us interpret our data and organize our results. And so, my collaborators are still, sort of these experiments are ongoing. And in fact, if you're interested in learning more about some of the experimental details, please reach out to my collaborators, including Luca Pinello, who's here today. So this is all ongoing work, so it's exciting, and we'll have more to say later. But let me just quickly go over some preliminary results, which at least suggest that doing this kind of Bayesian variable selection thing might be useful. So the context here is that of B-cell leukemia. The targeted is a putative enhancer of CD19, which is a gene. And CD19 is also a cell surface antigen found on most B-cell lineages, B-cell lymphomas and leukemias. It's a transmembrane protein, and it's known to play an important role in B-cell development. And so we used a single guide RNA library using the adenine base editor 8E to introduce edits into these cells, and followed the procedure on the previous slide. And these are the kind of inferences that we've gotten so far. And so all of this is preliminary. Well, let me just quickly sort of go over what we see here. So first of all, we have 3 replicates. Along the horizontal axis, we have genomic position. The blue dots correspond to the wild type, right? So if there are no edits, you could read off from the blue dots, what the actual underlying genomic region, what the sequence there is. But we're, of course, introducing edits. And since we're using a base editor, we can, there are at least in a world where the base editor could introduce all possible edits, at every position, there's sort of 3 possible edits you can make. And so that means that there are sort of 3 coefficients that we introduce at each genomic position that encode the sort of impact of making an edit at that site. And in practice, these base editors don't actually introduce, we don't actually get the full allelic diversity that we would hope, where we're really densely tiling and observing every possible edit, but we still see a fairly large number of edits. And so once we've run inference, then the model is going to try to explain the observed data, and particularly the observed differences between high and low, by using as few sort of coefficients as possible. And at least in this preliminary analysis, we see that so the sort of orange and red blobs correspond to those hits that we've identified that have large pips. So basically the size of those blobs is proportional to the pip. And you can see that at least in these preliminary results, there's at least one hit in the one particular region, sort of in the middle, that seems to be replicated across all 3 hits. So that would be, how we would use pips to rank our hits and identify a putative, part of this genomic region where we think that there's really, edits, particularly at that site, are causally linked to the target gene. And so that's how we would hope that, we'll see how this works on other data sets, but our hope is that you will see sort of a nice clean picture like this on other data sets, and how well it works in practice sort of still remains to be seen.
Do you expect adjacent sites to have similar effects? Like, is there a smoothness assumption that's at play here as well?
So we're not, you could have put an assumption like that, but we're not making that assumption.
It was maybe more a biological question underlying that. And then, is there a bias? And I guess it depends sort of on the model that you choose, whether it's a counting model or a normal model towards which sequences, like the representation of in the high and low bins of particular sequences. Like if there is a really large readout for one side in the binomial model, I feel like that would almost force the beta to be non-zero because it's going to have a very strong concentration around a non-zero value.
Yeah, so definitely one of the trickier things here is choosing what the right likelihood should be, right? You know, if we knew that the noise is actually binomial, then it would make sense to use the binomial likelihood, right? But the actual true noise is probably quite a bit more complex, and in particular is probably much more variable than that. And so we're still sort of investigating that. But you know, on the one hand, you want to take into account the fact that if you have more reads, that is more information. On the other hand, you don't want to maybe over trust the fact that you have a lot of reads at one place, because then you like you said, you might just get swamped by you know, the fact that certain areas are supported by more reads, and you shouldn't maybe take that too seriously. So there's sort of a balance there that somehow needs to be established.
I guess along similar lines, your likelihoods distinguish, I guess, because you have two things going on, right? You have expression, causal change in expression versus growth. So like,
What do you mean growth?
well, you could, for example, you could have a like expansion of a specific perturbation could have an effect on cell growth, and so that could like dilute. So you could get a huge number of reads in like, say you have a small effect on expression but a huge effect on growth, then you get like, you see that one you see that read like massively overrepresented. So there's like a difference between growth and expression, and I was wondering if that's incorporated in your likelihood at all.
Yeah, I don't know. I mean, we don't incorporate that explicitly, but in addition to sorted cells, in addition to sorting some of the cells are kept pre-sorted, and we sequence those as well, so we can sort of like look at different subsets of the data. And I don't think we've seen any evidence that would suggest that anything like that is going on.
And did you say, going back to the smoothness question, where adjacent sites biologically might be related, you said that you could incorporate it?
You could. I guess I'm always a bit wary to put in too many assumptions, given that there's just the worry that you'll just reproduce what your assumptions were. I'd rather have the data show me that. So, yeah. I guess if you're in a scenario where you might get better power by doing so, assuming it's a good assumption, but you also might bias yourself. So, what I would hope is that you have enough data that you can pull something out without doing that. If you ended up running a screen like this and you don't see any signal, then maybe you need to start thinking more about, maybe you need to make stronger assumptions or like filter your data more cleverly. But if at all possible, I'd like to get away with using the most generic assumptions possible. Okay, so that ends this second application. So let me just very quickly discuss this open source library that I created and just try to show to you that it's actually pretty easy to use and encourage you to use it if you're interested. So it has a few properties. One of them is that it supports both CPU and GPU workloads, which is useful because if it's a smallish data set, you should just use a CPU. But if it's a large data set, you probably want to use a GPU. Kind of the details of how scalable it is going to, of course, depend on how large the data set is and so on and so forth, or on your hardware and this, that, and the other. But in like very generic terms, it can handle data sets with tens of thousands of data points and about 1 million covariates. Details, of course, will change those numbers, but that just to give you sort of rule of thumb. It supports multiple likelihoods. So first of all, normal likelihood, a Bernoulli likelihood, and a binomial likelihood, and a negative binomial likelihood. And there's an open source GitHub implementation, which you can find at this link. It's well tested, and I think it's pretty easy to use. So let me try to prove that somewhat explicitly. So here's some code that implements Bayesian variable selection under the assumption of a normal likelihood. So how does this work? So in the first line, we just import this normal likelihood variable selector object. We take all our data, in other words, our covariates X, but also our response Y, and package it as a pandas data frame. We pass that data frame to this object. we tell the object which column refers to the response. It then assumes that all the other columns are covariates. And then you tell it, for example, what the expected number of covariates is a priori. There are different ways you can specify this. The simplest way is to just say the number of expected covariates a priori. And of course, remember, this is just a prior, this number controls a prior inclusion probability. That isn't saying that we're only going to look at models that have exactly 5 covariates, but it sort of sets, the overall level of expectation. And if the data needs to overwhelm that, it will do so. So it's not, necessarily such a large assumption. Once you've created this object, then you just run the MCMC algorithm for a given number of steps. And once you've done that, the sort of results of inferences are stored as various pandas data frames in this object, and you can sort of do whatever you like with those pandas data frames. And this is basically what that pandas data frame looks like for a sample data set. So in the first column, we just have the name of the covariate. In the second column, we have the estimated pip between 0 and 1. And the next column, we have the estimated posterior mean over the coefficient averaged over all models. In the next column, we have the standard deviation. And in the final 2 columns, we basically have the same sort of information, but these are conditional in the sense that it's conditioned on that model or that coefficient or that covariate being included in the model, right? So we have this huge space of models. Some of them include a given covariate and some do not. The second and third column here are averaging over all models, but the rightmost columns are just averaging over those models that include that particular covariate. So that tells you what's going on conditionally if you assume that that covariate is in your model. And that's it. So if you want to use it, check it out. You can follow this link or just search for it on GitHub and you'll find it pretty easily. Okay, so let me just very quickly summarize. I've tried to argue that Bayesian variable selection is a powerful method for inference in high-dimensional and generalized linear regression, and tried to show how that might be used in practice in bioinformatic applications. So both applications I talked about were in the sort of generic framework of trying to go from genotype to phenotype, which of course is a pattern in very many bioinformatic analyses. And so, generically, you might hope that doing something like this is useful in many such analyses, although of course it depends on the details of the analysis. Something that I think makes Bayesian variable selection particularly useful for these kinds of bioinformatic analyses is, first of all, that we're able to compute PIPs, or posterior inclusion probabilities, which are useful for ranking hits and other things. It can deal gracefully with highly correlated covariates. And if we want, we can incorporate prior knowledge, right? So for example, if I wanted to assume a priori that alleles in the S gene are more likely to be non-neutral, I could have done that explicitly in the model, and it would have sort of magically been incorporated in my posterior distribution. So if you want to include that kind of prior information, you have a sort of clean way of doing so. And importantly, sort of recent algorithmic and hardware advances make these methods more applicable to larger problems, right? So, depending on the size of your data set, it may or may not be applicable, but actually, there are many problems for which it actually is applicable, and it's actually pretty fast. And so if you're interested in trying this set of techniques on your data, check out Millipede, this open source package I just described, and you can find the link here at the bottom. And so I should acknowledge before I finish, people who are involved in this work one way or the other. Some of these people were sort of more directly involved. Some of them are just people I benefited from having conversations with. But thanks to all the people listed here. I hope I haven't forgotten anyone. And then I leave you finally with a number of references relevant to the applications I talked about, but also just Bayesian variable selection in general.